Thank you very much. It's an honor and a pleasure to give this seminar. I will start by sharing my screen so you can see slides. Let me know if you see it correctly. It's fine. So my group has been working in really to try to understand aging from the molecular point of view, because we think that aging is at the origin of many diseases, many of the non-infectious diseases, such as cancer, cardiovascular, neurodegenerative diseases, degenerative diseases of different tissues like lung, liver, kidney, fibrosis, metabolic diseases like diabetes. So therefore, we think it's very important to understand aging from the molecular point of view to be able to understand the origin of these diseases, and therefore to be able to prevent them or to treat them effectively. So aging, we think, is the cause, and the diseases are the consequence of this cause, which is this molecular process of aging. Aging can be influenced by the genes. Different species have different longevities, and this could be related to the genes, but also within single species, such as, for instance, the Homo sapiens, our species, the life habits can influence this speed of aging and could also have a role in how early these different diseases may be happening. In any case, if we know these molecular causes of aging, we will be able to have biomarkers to allow us to prevent or to diagnose early these different pathologies. But we should also be able to delay aging, and we should be able to design interventions that delay aging, so to be able to increase the health span of individuals and delay many different diseases at once, not only one disease, but many different diseases at once. So this would be something important that we could do if we really understand aging. And so what do we know about molecular aging? So there are many molecular pathways that have been involved in aging during the last years. And I will be talking about one of them, which is the shortening of structures that are at the end of the chromosomes. The chromosomes is where the DNA is located. And at the end of the chromosomes, there is a very important structure known as the telomere, which is important to protect our chromosomes, to protect our DNA. And we know that this is, we think, is a primary cause of aging, because when telomeres are eroded, and this is something that happens associated with life. So as we age, our tissues have to be regenerated, and this is leading to a progressive telomere shortening, because telomeres shorten associated to cell division, to cell multiplication and regeneration. And this telomere shortening will lead to other causes of aging, like genomic instability, stem cell exhaustion, cellular senescence, mitochondrial dysfunction, and in general, we think maybe triggering many different causes of aging. So that's why we think it's a primary event, this shortening of telomeres that happens associated to the regeneration of our cells and our tissues. There is an enzyme, it's called telomerase. Telomerase is able to re-elongate telomeres. In fact, telomerase is present at the early stages of embryonic development, and there telomerase is activated, and it can elongate the telomeres of the new individual. And basically, after that, telomeres start shortening, even during the embryonic development. When we are born, telomeres can shorten. And we think this is at the origin of aging. So if we measure telomere length versus the age of an individual, the telomeres shorten in all organs and tissues, and this also includes the stem cells. And when telomeres are too short, we think this is at the origin of disease. Cancer is an age-related disease. It also happens associated with aging. But in the case of cancer, there is a unique property of cancer cells, which is that they are able to divide indefinitely. And this is because they can reactivate telomerase. And this happens in the majority of cancers. It's one of the most mutated genes in cancer, telomerase. Therefore, also demonstrating that telomere length is really limiting for the ability of cells to divide. And for instance, cancer cells can divide beyond what is normal by reactivating telomerase. So this is pointing to the importance of this enzyme in maintaining telomeres and allowing for cell division. We know that there are a number of diseases which are called telomere syndromes that are associated to mutations in telomerase. So these individuals have abnormal telomerase activity, telomeres are not properly maintained, and they are going to prematurely develop a number of diseases which are characterized by the premature loss of the regenerative capacity of tissues. So this is telling us that the telomere length is rate limiting for human longevity, and if you have less telomeres than normal, you are going to develop prematurely a number of diseases which are characterized by the loss of regenerative capacity of tissues. Even in individuals that have a normal telomere length, that have a normal telomerase gene, we know that different habits can influence the rate of telomere shortening. It has been proposed that telomere length is one of these biomarkers of aging. Shorter telomeres than normal could be really a prognostic factor for different diseases, like cardiovascular disease, for example, and other diseases, cancer as well. So we think that this telomere length could be a biomarker of aging. We know that telomeres are very important even before the human syndromes. I participated together with a group of Carol Greider in demonstrating that telomeres are at the origin of aging by generating a telomerase deficient mouse model. So these mice have abnormally short telomeres, and we could demonstrate that short telomeres were sufficient to induce chromosomal instability, to induce the loss of the regenerative capacity of tissues, and to induce many different degenerative pathologies in mice. This was, as I said before, the human syndromes were discovered. Also, these mice, because they don't have telomerase, they are resistant to cancer. We try to induce cancer, which is also showing the importance of telomerase for cancer growth. So a cancer cell cannot grow if it does not have a mechanism to maintain the telomeres. So telomeres are important in humans. They are important in mice. However, there was something interesting. We humans are born with very short telomeres compared to mice. So mice are born with longer telomeres than humans, but we can live up to 80 years and a mouse can only live 2 years. So some people thought, well, this means that telomeres are not determining aging because a mouse is born with very long telomeres but has a very short life. My group and then also other groups have shown that is not the initial telomere length what is important, but the rate of telomere shortening. We found that mice shorten their telomeres 100 times faster than humans. So probably this is why, even though they are born with long telomeres, they live much shorter because the rate of telomere shortening is much higher. We more recently showed whether this was true for other species. So we want to understand whether telomeres are determining longevity in many different species, not only mice or human. And we published a study in which we measured telomere length in different species of mammals and birds, and we determined the rate of telomere shortening, and we determined telomere length. We saw that telomere length at birth was not predictive of longevity, but the rate of telomere shortening was predictive for longevity. So we have measured telomeres by technology that we developed, which can measure telomeres in individual cells. We measured telomeres in many different species, and we measured the rate of telomere shortening. And we saw that the rate of telomere shortening was predictive of the lifespan, and it could be adjusted to power law, which is a mathematical equation that can explain longevity or lifespan by using the rate of telomere shortening. So you can have two species, like the elephant and the flamingo. They are very distant in evolution. So the elephant is a mammal, the flamingo is a bird. However, they have the same rate of telomere shortening, and they also share the same longevity, more or less. A flamingo or an elephant can live up to 60, 70 years of age. An elephant and a mouse, they are both mammals. They are much closer, genetically closer. However, the mouse only lives 2 years, an elephant can live 70 years, and we think this is because they have very different rates of telomere shortening. So telomere length, we think, is very important to determine longevity, not only in mice and humans, but maybe in different species, like we are seeing here. So while the telomeres are important for aging, can we delay telomere shortening? Can we show that really telomeres, if their shortening is delayed, we can increase longevity? We did this in 2008. So we generated transgenic mice that we were able to maintain their telomeres longer for longer times. And we could see that the mice that had longer telomeres, they had an increased median survival of about 40%. And they also had an increased health span. So the first mouse that die 3 times later than in the normal mice. In the normal mice, the first mouse die at a year of age, more or less. But in the group that maintain telomeres for longer, the first mouse die much later. So we are in delay in aging. And we showed that we could delay many different pathologies. In fact, at 3 years of age, the majority of the control mice were dead, but the mice with long telomeres were still alive. And you can see here an image of 2 mice which are chronologically very old, 2 years old. This looks like an old mouse, but this one with the long telomeres looks like a younger mouse. So we are delaying aging, and with this we are delaying pathologies associated with aging. We cannot do human transgenics, but we can think about therapeutic strategies to activate telomerase, which could elongate the short telomeres and maybe prevent, cure diseases associated with aging. With this in mind, we tested a telomerase gene therapy using adeno-associated viruses. So these viruses are non-integrative, and they have been used to express genes in humans to cure genetic diseases. So we thought that aging could be also due to the lack of telomerase in the adult organism, which causes telomere shortening. So we would be able to activate telomerase and deliver telomerase into an adult organism. Maybe we could delay aging, delay age-related diseases. So we did the experiment with mice that were middle-aged, 1 year of age, or mice that were old, 2 years of age. And we give them a single intravenous injection with these gene therapy vectors. And we saw that the mice that were treated with telomerase, they had longer telomeres, they had less DNA damage as they were aging. And we saw that we could delay many different pathologies associated with aging, including cancer. So cancer was also appearing later. And this means that cancer is also caused by aging. And if we delay molecular aging, we can delay cancer. And we did not see any negative effect of expressing telomerase in this way, because the mice actually live longer. The 1-year-old group lived 20% longer, and the 2-year-old group lived 13% longer. So we do not see any deleterious effect of activating telomerase by using gene therapy. So what we have done after that is, well, we don't want to increase longevity. This is not the priority. The priority would be to have treatments for age-related diseases, which we don't know how to cure right now. There are many age-related diseases that have no effective treatment. And what we did was to generate mouse models that recapitulate these diseases in mice and try to cure the different diseases by using the telomerase gene therapy. We showed that in mouse models of heart infarct, the telomerase gene therapy had a therapeutic effect. We also saw that a therapeutic effect in mouse models of aplastic anemia, which is one of the telomere syndromes. And more recently, we have shown also that in pulmonary fibrosis, and I will be talking more about pulmonary fibrosis,